Alright guys, so now I'm going to try to do a demonstration using the socks that you should be slightly familiar with. Sorry for the weird angle on the camera. It's the best way I could think of. Okay, we're starting off with, we're going to say this is the dad. Okay, the dad is homozygotic dominant. So capital F, capital F. He's homozygotic dominant for this gene too, as well as this gene. So he's a purebred homozygotic dominant creature. Okay, so we're going to go back a little bit to the mitosis and meiosis, and we're going to review that, and we're going to kind of try to put it all together right now. So right now we're in interphase, where the genes are just chill, they're chromatin, and they're going to start doubling up. Okay, so now we've made copies, and the genes are all good up in the nucleus. Now we're going to go into prophase. Okay, prophase, so we are matching up into sister chromatids. Okay, so notice how they doubled up, they copied themselves, and you've got two F's, two capital F's. Your ends are copied. Okay, ends are copied. Prophase. B's are copied. B's are copied. Okay, and after prophase, we go to, remember, prophase, prophase, metaphase, metaphase, where we line up on the metaphyseal plate, and then anaphase, anaphase, they get pulled apart. Okay, telophase, telophase, and cytokinesis. Okay, in cytokinesis, we can take off the centromeres. We've got two cells with exact copies of the DNA, right? At this point, this is mitosis, and we are going to go into meiosis. So this would be mitosis 1. So now each of these cells is going to split again, but we're not doubling up our DNA. So we're going to go metaphase, lining up the socks on the metaphyseal plate, or lining up the chromosomes on the metaphyseal plate. Okay, then anaphase, 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 and now, well, telophase, cytokinesis. Now we have 4 cells that each have one copy of the genes from that original cell. And you'll notice they're all the capitals. Okay, they're all dominant because this creature was homozygotic dominant and he didn't have any other genes he could give off. Right? All right, now we've got our sex cells, our gametes. So these are the potential cells that could fertilize an egg. So now we're going to move the dad's potential gametes over here, and we're going to bring in the mom. Now this mom is homozygotic recessive, okay? Meaning she's got lowercase b, lowercase f, and lowercase n. There we go. Okay, so she's homozygotic recessive. And so in interphase, her DNA is just hanging out, and then it doubles. Okay, woo, all doubled up. Now it's going to pair up into its sister chromatids. Okay, we go through mitosis 1. Okay. Pairing up. Pairing up. pairing up. And pairing up. Okay, so we've got the copies, and this is lowercase, 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 because that's all the mama had to make copies of. Lowercase, 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 and once again, she's homozygotic recessive, so she can only have copies that are recessive, okay? So these are all mixed up, and it goes into prophase, and then after prophase, we go into metaphase, lined up on that metaphyseal plate. After metaphase, we go into anaphase, separating them out, because Anna's such a spiteful girl. After anaphase, we go to telophase and cytokinesis. And we have two daughter cells that are diploid, so exact copies of each other with all of the DNA, right? All right, now we go into mitosis 2, or meiosis phase. Okay, so that's going to be where the chromosomes are all happy, right? They're all woo, chill. And they go into their centromere paired chromatids, sister chromatids, okay? And there we go. So we have our two daughter cells that are original copies of that first cell, okay? And remember, now we don't double the DNA, okay? But we do line up on that metaphyseal plate, okay? So each one is its own cell, and they're lined up on that metaphyseal plate, okay? After metaphase comes anaphase. After metaphase, ouch, comes anaphase. Just snapped myself in the finger with a rubber band. Okay, so let's separate them out. All right. Then we are at telophase and cytokinesis with the 4 daughter cells that all have the exact copies of the original cell's DNA, but only half of them. They're haploid. Okay? All right. Just proving to you that I haven't snuck anything to trick you in here. Everything you see is exactly what you get. All right. So now we've got the 4 cells of the mom's gametes, the 4 mom sex cells with the genes that she can potentially give to an offspring. All right? All right. So with the mom, it's all homozygotic recessive. With the dad, it's all homozygotic dominant. Okay? So it doesn't matter which sperm fertilizes which egg. All of the genetics are going to end up being exactly the same, because all the genes that the dad can give off are dominant, and all the genes that the mom can give off are recessive. Okay? All right, so if this were, we're not talking about any phenotypes right now, we're only talking about genotypes, so I want you to know that all of these genotypes are recessive, all of these genotypes are dominant. Feeling comfortable with that? Okay, then let's do this. This egg gets fertilized by this sperm, okay? Rest we can set aside for now. We are only worried about this egg and this sperm, okay? So now we've got the dad and the mom's genotypes combining, making a heterozygote. 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 So how did that happen? Well, the dad's gene and the mom's gene were here. Okay? They combined like that. If you want me to show you, or, hold up. Okay. So if we were going to look at this as in a Punnett square, we would say, okay, here is the dad's potential. Capital F, right? That's all he can give off for this particular gene. So we're going to put those right there. And all he can give off for this gene, capital B, put those there. And all he can give off for this gene is the capital N, okay? Okay, so now I'm going to do Punnett squares with the socks. First we're going to do the orange, then we're going to do the black, then we're going to do the green. For the mom, the only genes she has to give are the lowercase. So I'm going to put those, let me see, move this back a little. Right here, see where they are? Okay. All right, for the mom with the B genes, recessive, okay, and the mom with the N genes, recessive, okay. All right, so I'm going to move these guys over for just a minute so that way you can see the orange ones a little bit better. Okay, we have our Punnett square. Dad on top, mom on the side. Okay, dad can give a capital F and a capital F. Okay, that's coming from here down into this box, down into this box. And then dad can give from this box a capital F and a capital F. Okay, so drop down, drop down. Now mom, we're going to slide over the lowercase f's. So here, lowercase f, sliding this over, lowercase f, sliding this over, lowercase f, sliding this over, lowercase f. All right, so now here are the 4 quadrants of our Punnett square. This is the dad's genes, this is the mom's genes. So you can see here we've got a heterozygote, capital F, lowercase f, capital F, lowercase f, capital F, lowercase f, capital F, lowercase f. Those are the only genetic combos that can be made from the 2 homozygotic dominant and homozygotic recessive parents. Okay, so all of their offspring will have a 100% chance of having the dominant phenotype but being heterozygotic. Okay? All right, so let's take, these are 4 possible phenotypes. Okay, we're going to put those up. Okay, we're going to move these guys here because that's 1 gene and we've got 3 genes to do. All right, dad, homozygotic dominant, mom, homozygotic recessive. Okay, and homozygotic dominant from daddy, homozygotic dominant from daddy, homozygotic dominant from daddy, homozygotic dominant from daddy, homozygotic recessive from mama, homozygotic recessive from mama, homozygotic recessive from mama, homozygotic recessive from mama. Okay, so mom and dad have their DNA into these potential Punnett square quadrants. And so once again, you've got homozygotic dominant and, a heterozygotic dominant, heterozygotic dominant, heterozygotic dominant, heterozygotic dominant. So these are the 4 potential genotypes for their offspring, which again is 100% heterozygotic dominant. Okay? All right? Now for the N's. Capital N, lowercase n, okay? Capital N from daddy, capital N from daddy, capital N from daddy, capital N from daddy, lowercase n from mama, lowercase n from mama, lowercase n from mama, lowercase n from mama. Mom and dad's possible genetic combination is once again going to be a heterozygote dominant. Okay, 100% heterozygote dominant. All right, there we go. So all of the offspring will end up being heterozygotes. Okay, so the offspring for their kiddos, or the genotype for their offspring is going to be heterozygotic, heterozygotic, heterozygotic. And no matter how many offspring they have, it's always going to be the same heterozygotic. 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 I put 2 here. Okay. So now let's look and see what the next generation would look like. If you have, this would be the parental generation or the F1 generation that generated these guys. Okay, now we've got our heterozygotes, okay? And daddy has the ability to give a capital B and a lowercase b. Okay, we're going to start with the blacks. Mommy has the ability to give a capital B and a lowercase b. All right? Okay, now capital B comes from daddy. Capital B comes from daddy. Okay, lowercase b comes from daddy. Lowercase b comes from daddy. Capital B comes from mommy. Capital B comes from mommy. Lowercase b comes from mommy. Lowercase b comes from mommy. Parental generation genetics are done. There are possible genotypes that they can give off. Homozygotic dominant, heterozygotic, heterozygotic, and homozygotic recessive, meaning that you have a 25% chance of being a homozygotic dominant from their F3 offspring, a 50% chance of being homozygotic recessive, but will show, heterozygotic dominant, but it will show the phenotype of the dominant, and a 25% chance of being recessive. So the grandparents might see a child, an offspring, their grand offspring that looks like the grandmother, okay? But she won't have it in her kids. She has the potential to see it in her grandkids, okay? All right. So we're going to take these guys and put them over here, okay? Remember, this is the F2 generation, or the F3 generation, one of their genes. Now, here is the ends for the daddy. Remember, he This is the F2 generation. Okay, he is heterozygotic dominant, and mama is heterozygotic dominant. Okay, so daddy can give off from here a capital N and a capital N, and from here a lowercase n and a lowercase n, and mama can give off a capital N, and a lowercase n, whoop, there we go, lowercase n. All right, we're done with the F2 generation. We're looking at F3. F3, you've got a homozygotic dominant, a homozygotic recessive, and a heterozygotic, two heterozygotic dominants. Okay, once again, meaning that you're going to have a 75% chance of the dominant phenotype with a 25% chance of the genotype being homozygotic dominant, and a 50% chance of the genotype being heterozygotes, and a 25% chance of a homozygotic recessive phenotype and a homozygotic recessive genotype as well. Okay, remember, phenotype is what you see, genotype is what you don't. Now I'm just going to put these offsprings, the F3 generation over here, and we're going to do the oranges. Okay, daddy can give, our daddy is heterozygotic dominant, mama is heterozygotic dominant. Okay, all right, so daddy can give a capital F, a capital F, a lowercase f, a lowercase f. Mama can give a capital F, a capital F, a lowercase f, a lowercase f. All right, we're done with those two parents. And we have the potential genotype for the offsprings, okay, being homozygotic dominant, heterozygotic dominant, heterozygotic dominant, and homozygotic recessive. Okay, 75% chance of the dominant phenotype with a 25% chance of the homozygotic dominant genotype, a 50% chance of the heterozygotic genotype, and a 25% chance of the homozygotic recessive phenotype, as well as a recessive genotype. Okay, so now I'm going to take these guys Okay, here are the potential genotypes for the F3 generation. We're going to make an F4 generation, and we are just going to mix these suckers up. All right? All right. Law of independent assortment. Here is 1 offspring, 2 offsprings, 3 offsprings, 4 offsprings. Okay? All right, everybody with me? This offspring ended up being homozygotic recessive for this gene, homozygotic recessive for this gene, and homozygotic recessive for this gene. That was just random, okay? This offspring, heterozygotic or homozygotic dominant, heterozygotic dominant, and heterozygotic dominant. All right. This offspring, it is heterozygotic dominant. Heterozygotic dominant. Heterozygotic dominant. Okay, that's the third offspring. And the fourth offspring happened to be heterozygotic dominant. Heterozygotic dominant. Heterozygotic dominant. All right, so now what I'm going to do is I'm going to mix these offsprings up so we don't know which are going to mate with which. And if you are able to keep an eye on what's going on here and realize that I cannot do sleight of hand. Okay, these guys are going to have babies. These guys we're going to set aside. Okay? All right. This is going to be the daddy. He is homozygotic recessive. This is the mommy. She is heterozygote. Okay? All right. So, the daddy can only give on this one, okay, little f, little f. Okay, here we need 2 more little f's. Little f. Oh, I'm out of little f socks. That doesn't make any sense. Okay guys, don't worry, I got this. Little f. Okay, all right. Okay, now the mama can give a capital F and a capital F. Okay, and I'm still out of little f socks, so little f and little f. All right, so now the parents go away, and here is the potential offspring, capital F, lowercase f, heterozygotic, capital F, lowercase f, heterozygotic, lowercase f, homozygotic recessive, lowercase f, homozygotic recessive. So this generation, you have the potential of 50% heterozygotic, 50% homozygotic recessive. Okay? Now we're going to put these guys over to the side, and we're going to do the B's. Daddy has heterozygotic dominant. Mommy has homozygotic recessive. Okay, so can you see how the genes can go through many potential lineups, many different generations, how you can see some traits that occur in every generation because they're dominant, and some that occur in only a very few generations because they're recessive. Okay, that's the whole idea behind this. The law of independent assortment and the luck of the draw. So, and the law of segregation. That one shouldn't be there. This one should be there. And I am out of little B's. So, and you just don't know what you're going to get. You have an idea because you have a gene pool, okay? You have an idea because you have a gene pool. But you don't know for sure unless you are working with a pure homozygote dominant or pure homozygote recessive. If you are working with a pure homozygotic dominant or a pure homozygotic recessive, you know the only genes that they can give off. So some of you are going to ask, well, how do you know if you've got a pure homozygotic dominant or recessive? With the recessive, it's easy. You know you've got a pure homozygotic recessive because you can see it. If you see a recessive phenotype, then you have a recessive or a homozygotic recessive organism for that, or in their genotype. If you see a homozygotic dominant type, you could have a homozygotic dominant or a heterozygotic dominant. You don't know. So you'd have to go through multiple generations of breeding them to find out. Or you could run a genotyping. You know, you could do that. Genotyping is not always in everybody's budget. So that, whoops, that makes it not accessible to everyone. FFBB, FF. Somebody's missing a B. BB, B. Ah. Okay. All right. And N, and N, and N, and N. You don't know how they're going to come out. So now you've got this offspring, heterozygotic, homozygotic recessive, heterozygotic. All right. Heterozygotic, heterozygotic, heterozygotic recessive, heterozygotic, heterozygotic, I'm sorry, homozygotic recessive, heterozygotic. And the last one, homozygotic recessive, homozygotic recessive, heterozygotic dominant. Okay. I hope that made sense to you guys. I am going to run through just one more time a homozygotic dominant cross with a homozygotic dominant, and then a heterozygotic or homozygotic recessive cross with a homozygotic recessive. So recessive, recessive, dominant, dominant. Okay? Dominant, dominant. Okay? Daddy can give a capital F. Daddy can give a capital F. That's all the capital F's I have, so now Daddy can give a capital F, capital F. Mama can give a capital F, capital F, capital F, capital F. Okay? And so what you have here for the offspring is 100% homozygotic dominant. 100% homozygotic dominant in your genotype as well as your phenotype. Okay? Now that's going to mean that if you do that for a homozygotic recessive cross with a homozygotic recessive cross, you're going to get the same thing. Lowercase f, 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 lowercase f. So they're called purebreds because the F2 generation will show the purebred non-dominant gene, recessive gene. Okay? All right. Practice, play, have some fun. Look at the other things that I've posted. Check out all of that. Please don't do the assessment until after you've looked at everything. Please do not start off with the assessment and then tell me that you don't understand anything that's going on. Remember, your book, your partner, your book, your partner, me. In this online world, it is all of the resources that I have provided for you, your book, the PowerPoint, the lecture, me. I have no problem helping you navigate through all of this mess. I have no problem answering any and all of your questions as long as you are utilizing your resources. Just like in the classroom, you've got to read first because I don't want to type in the same answer 25, 45, 65 different times when it could be seen when I've posted the answer on the stream or when I've given all of the information that is required, needed to help you be successful in your classroom assignments, okay? I love you guys, I miss you guys, and I can promise you that I am not meant for online teaching. I am meant to be in this classroom, working with your personalities, bouncing off your energy, making the day happen fast and good and laughing. Not lecturing to an empty classroom. Well, I'm lecturing to the hand sanitizer bottles. I don't know if that really counts though. Love you guys, miss you guys. Ask questions if you need to. All right, bye.